back, folks. Today's talk is going to be about the prokaryotes. So we're looking specifically today at cells that don't have an actual nucleus inside of them. Instead, we have loose genetic material on the inside. Typically, they don't have cellular organelles that we're used to when we study cells. These are really basic small cells, much smaller than the eukaryotic cells that we're most used to studying. And with prokaryotes, we're going to be covering the bacteria and the Archaebacteria. So the bacteria are in one domain, Archaebacteria or Archaea are in another domain, and then eukarya are in the third domain. And eukarya covers things like plants, animals, and fungi. So let's take a look at what we want to know by the end of today's lecture by way of our learning targets. Number one, we want to be able to explain how some bacteria are classified. Number two, describe structural characteristics of a bacterium. 3, describe functions of those structures. 4, explain how bacteria reproduce. 5, describe mechanisms of genetic change in bacteria. 6, explain what spores are. 7, describe why bacteria get a bad rap. And 8, explain the sneaky mechanisms that allow bacteria to survive. Let's get to it. Let's start off by going over some general characteristics of prokaryotes, starting with the idea that unlike viruses, bacteria and archaea are fully functioning cells. So we just got through studying viruses and we learned that they needed to have a host cell present in order to do what nature has designed them to do. But now that we've gone up one level, we're now at the cellular level where all of the structures that require functioning within one particular entity, one cell, are all done. And bacteria or the prokaryotes are able to do that. They're found in nearly every environment on Earth. So whether it's a swamp, whether it's a desert, whether it's an icy field in Antarctica, we find prokaryotes everywhere on Earth. They are masters of adaptation. We're going to learn later their mechanism for changing not only their structures, but how they function through nothing more than interacting with one another on a regular basis. Bacteria have existed for over 1 billion years on primitive Earth. Imagine that for 1 billion years on our hot, rocky planet. Nothing but bacteria around. They're very small. They range in size from 1 to 10 micrometers in length and can be 0.7 to 1.5 micrometers in width. If you take a look at the 3 pictures on the right of your slide, you can see that bacteria typically take 3 basic shapes. The first shape is called cocci, and these are kind of roundish or elongated and roundish looking structures, but cocci are the most round of the bacterial shapes. We also have bacilli, which are a little bit more rod-shaped. Now, if you look at the little seam going down the center, you can see these are actually in division right now. So we have, for example, these two are going to be separating in just a moment when they pull away from each other. Also, these, you can see kind of a singular round coccyx right here, and here you see kind of a double structure because this one as well is going through cellular division. The third type is called sporili, and you can see it kind of looks like a little worm. It is motile, so it can move around. So these are one of the ways in which we can classify bacteria based on their shape. So we have round, rod-like, and spiral. Bacteria played a role as we tried to disprove the idea of spontaneous generation. Spontaneous generation was when living things arose from non-living things. So Louis Pasteur, through a very simple experiment, was able to go ahead and show and disprove that spontaneous generation actually happened. So what he was going to do is basically take a flask that was full of microbes, and he would apply some vigorous heat to it, and by boiling that liquid broth full of microbes, which would include bacteria, he'd essentially kill them all and make this resulting liquid sterile. So after he did that, he now has a sterile broth free of live cells. Once it's been boiled for a given amount of time, there'd be nothing living left in it. And he went ahead then and he broke the neck off of one of his flasks and he left the other neck in place on a different sample. So by breaking the neck off of this one, it allowed for a hole for entry. So anything that was in the air could then go ahead and float down, perhaps riding an air particle down into the broth of the open top flask. Meanwhile, on what we call the swan neck flask over here, he didn't break it off, which means that anything that entered the open hole at the end would then fly in, but would be unable to crawl up this part of the swan neck flask to get to the broth on the inside. So instead, these little particles here carrying the microbes would land and they would get stuck right here. And after a given amount of time, the sterile would remain completely microorganism free. Whereas the one over here, by allowing those little microbes to fall into this nutrient broth here, this would become very cloudy and full of living organisms within days of it setting out on a lab counter. By doing this, Pasteur was able to show that life does not just appear spontaneously, but instead all life comes from other life. Let's look at some structural basics of prokaryotes. Let's start with the fact that all prokaryotes lack a membrane-bound nucleus. So again, we said that there was no actual nuclear membrane and that the DNA was just kind of free-floating within the sample itself. And this right here is your bacterial DNA. Now, you can see it's kind of all clumped up and folded every which way, but if you were to unwind it, this would actually be a ring of DNA. So we call this area where we find the DNA the nucleoid region. Bacteria also have outer cell walls that contain a chemical called peptidoglycan, and that particular chemical will prevent the bacterium from osmotic bursting. So in other words, if water was to find a way into this bacterial cell, it would have a way of not exploding due to the incoming water into the cell. Because typically a lot of bacteria are going to find themselves in aqueous environments. And when a bacterial cell has a higher amount of concentration on the inside, water will try to get in there. So if it does, it's okay if you have a cell wall. And you can see right here, this middle layer, this is the bacterial cell wall. You can see a couple layers on either side of it, which we'll get to in a second as well. Some bacteria will move by way of flagella. So this is a singular flagellum right here. You can see it's a whip-like tail. You can see it's anchored right here through all three layers at this structure called a basal body. And it just basically whips around in an undulating motion, and that will propel the bacteria in a forward direction, which would be that way. Bacteria also lack membranous organelles. So if we look in here and we start looking for things like endoplasmic reticulum and lysosomes and Golgi apparatus, we're not going to find those things because they're much simpler than that. Bacteria may have accessory rings of DNA called plasmids. Now, if you look right here, we see this green ring inside of the bacterium. This is some extra DNA that's different from the DNA that essentially codes for this bacterium's existence. It's an extra set that it can actually share with other bacteria around it when we talk about that a little bit later during this lecture. We also have something called fimbriae, and these are these little tiny tentacle-like extensions right here that allow bacteria to attach to host cells and perhaps put a good grip on them while they're either infecting them or perhaps exchanging information with them. There's a lot of possibilities there. If you look at the electron micrograph down here, you can see there's kind of this real fuzzy border around the bacterium itself, and then you see these pink things here. These actually aren't the fimbriae. These are going to be what we call pili, or singularly we call it a pilus. And one of these little tentacles here can exchange genetic information to cells around this particular bacterium, and we'll explain how. Lastly, a capsule or slime layer can also surround our cell wall, and this will have carbohydrate chains protruding from it itself, and those carbohydrate chains may add to the pathogenicity, how harmful a particular bacteria might be, or it may provide a resistance or defense to certain drugs or chemicals that would normally kill it. And you can see right here that it's outside of the cell wall we were talking about. So it's this peach-colored layer on the outside here with all the spikes attached to it. So let's break down our prokaryotic structure. So within a prokaryotic cell, we'll find a cell envelope, also known as the cell membrane. We'll find cytoplasm on the inside. It has appendages. The cell envelope itself is made of the capsule, that layer on the outside we were just talking about, the cell walls underneath that, and it even has its own plasma membrane. So the three of those together are sometimes referred to as the cell envelope. Sometimes when we study eukaryotic cells, we'll actually interchange that term when we just talk about plasma membrane. But for a bacteria, those three are in such close proximity, we can collectively call them an envelope. The cytoplasm will contain that nucleoid region that we spoke about with the DNA inside of it. It will also have ribosomes, so it does build proteins regularly and all the, with every instruction that's coded on the DNA. And within the cytoplasm of some of our cyanobacteria, those that photosynthesize, we have what are known as thylakoids. Thylakoids we first learned when we studied photosynthesis. Some of the appendages that we find on bacteria are flagella, those whip-like tails that allow for propulsion, conjugation, pilus or pili, and those are going to allow for exchange of genetic material, and then fimbriae were kind of the little tiny finger-like projections that allow them to grip and grab onto other cells around them. Prokaryotes also have mechanisms for reproduction. Their key mechanism is asexually by means of what's known as binary fission. So let's take a look at the 3 steps that do that. Number 1, chromosomes replicate as the cell grows. So here you can see a bacterial cell. It is quite elongated from end to end. So that may be the trigger for there may not be enough resources if it keeps growing, so it will be more efficient if there's 2 bacteria instead of 1. So that might be the little tiny physiological trigger that tells that bacterial cell to copy the DNA. So here you can see the red and the purple, and they're kind of in the same shapes, shadowing each other because they're splitting from one another. Number 2, the genetic material separates and the cell elongates. So by the time we get down here, you can see that each copy went to either end of the cell, and we can already see a little bit of the folding inward of the cell membrane down the middle. So we have further elongation. And then in number 3, cross walls form and separate 2 cells. So right down the middle right here, we're going to be forming basically some new cell wall. The cell membrane will then line the inside of it, and if it has a slime layer on the outside, it will also be coated in that. So we just went from 1 to 2. Basically grow and divide, grow and divide, grow and divide is how they replicate so efficiently. Now another thing we should add in here is that all bacterial cells are haploid. Now, what that means is that they have a single copy of DNA. And if you have a single copy, whatever's on that DNA, that is going to be expressed within that cell and to that cell itself. Whereas diploid organisms, where there's 2 copies, you have issues of dominance and recessiveness and interaction of genes that may not express every trait that's coded for. Now, if a bacteria goes through a mutation, it instantly means that that bacteria is now mutated for all future lines because that mutation is now part of this DNA somewhere. So, for example, if there's a mutation right here on this particular chromosome, now you're going to split it and you're going to have that mutation as part of this bacterium here and this bacterium, and it will just continue carrying that new mutation onto future generations. They're also immediately subjected to natural selection. So what that means, if that mutation is beneficial, what we should see then is an increase in the amount of the bacteria. If the mutation is harmful, what we should see is a decrease because it reduced its fitness, it reduced its ability to adapt to its current environment. Let's take a quick look at how bacteria change over time. So these are going to be methods of genetic recombination. So if you look over here at the picture on the right, you can see there's two bacteria, and you can see this kind of wiggly red line between the two. Here you can see we basically have some fimbriae sticking off it, but this one here is really long. So this is what we call a sex pilus. And a sex pilus is literally a tube that goes from one bacterial cell to another. And what it's going to do, it's going to pass DNA from a donor cell. So this here will be our donor cell. This one here will be our recipient cell. So the recipient will receive that particular DNA that comes from a plasmid on the inside. Remember, plasmids are little round, extra DNA aside from its normal chromosomal content. So it might have like a little secret on this cell on how to get these great fimbriae on the outside right here, because this is a real successful adaptation for this bacteria. So it will pass on some of this DNA in this ring right here that codes for it to its recipient cell over here on this other side. And maybe that little bit of DNA will now cause fimbriae to start growing out of this one here. So it's a sharing mechanism of information. And by doing that, you basically have genetically recombined. You've taken DNA from this one and made it part of this cell over here. The second way that we can have genetic recombination within a bacterial cell is when a bacterial cell dies, it tends to spill out all of its contents. So let's imagine that something happened in this bacterium, its DNA got all chopped up, and there's some bacteria nearby that are ready to accept and bring in these little DNA segments and add them to its own ring-like structure of DNA on the inside. So perhaps there's a gene on this one here that it didn't have before until it absorbed what this cell that died spilled out of it. So we've transformed this cell here into something new using that extracellular DNA. So it's basically the picking up of extracellular DNA. The last method of genetic recombination is going to be through what's known as transduction. And this is going to utilize those phages we learned about during our virus chapter. So essentially we said that bacteria could be infected by viruses as well, and it's done through something called a bacteriophage. So let's imagine here that we have a bacteriophage virus landing on top of this bacterium, and it's injecting its phage DNA inside of this bacterium here, which already has gene A plus and gene B plus. Now, one of the things that can happen when there is a viral injection is there's enzymes called restriction enzymes that will go to work chopping up all the DNA in response to being infected. So let's imagine that happens here, and we can see all the DNA in the ring is being chopped up. We can see that we have injected phage DNA, which looks like it's already starting to become copied. And by taking over, this bacterial cell has become basically a virus factory. And you can see 3 out of the 4 that we have shown here are carrying that same viral bacteriophage DNA. But now, some of the original DNA, the segment that contained the A plus set of genes here, or gene itself, is also inside of one of the viral bodies and is going to be then passed on to a new cell. When that bacteriophage lands on a new cell and injects its contents on the inside, that content included the A plus gene. So now this DNA here will become part of this recombinant cell, which had the A minus and B minus gene, but now it also has the A plus gene. So whatever proteins that this codes for will now be expressed in what we call this recombinant cell. Bacteria endure all kinds of conditions, and sometimes when the conditions get too challenging for survival, some bacteria will form resistant spores under those unfavorable conditions. So what will happen when a bacterial cell forms a spore is the cytoplasm will dehydrate. Dehydrate means all the water will come out of it, and a copy of the bacterial genome is saved. So ultimately the DNA is still on the inside, but take a look. Suddenly you have multiple layers of things on the outside that are going to keep this DNA protected. So right outside the DNA, we have the cortex covered with what's known as the spore coat. And we even have a little bit extra on the outside here called the exosporium. So these things here are going to allow for this DNA collection to be preserved indefinitely until external conditions get better. Let's take a look at a really common one that we talk about when referring to spore formers. So there's this bacteria called Clostridium, and this family of bacteria, one of the diseases it causes is tetanus. You'll find Clostridium family bacteria in soil, saliva, dust, and manure, but you can also find them on rusty metal. And these bacteria can enter your body through a deep cut, like those you might get from stepping on a nail or through a particular type of burn. So normally this family here, they're going to live under anaerobic conditions, which means that they don't like air very much. But if they find a way to get inside your body, so let's say that this is a spore, and you step on a nail, it goes in your foot, and suddenly it has this warm liquid medium around it. It's going to go ahead and use that warm, inviting environment to almost spring back to life, to rehydrate the cytoplasm, almost like planting a seed in some wet dirt. That moisture is going to seep into the actual seed itself, rehydrating it and beginning the process of almost like coming back alive from a state of suspended animation. So here you can see you have a dormant bacterial spore covered with its spore coat. Add a little bit of moisture, a little bit of warmth to some of the different members of that group, and it will germinate back into active live bacteria. If you were to do this and you were to get a tetanus infection, what it does is going to basically, once there's enough of these bacteria inside of you, once they multiply, they can cause painful tightening of your muscles all over your body, even the locking of the jaw, which we refer to as lockjaw. So a tetanus infection can be very frightening for those that get it, and ultimately we want to see a doctor very quickly if we were cut by something like a rusty piece of metal, because there is that possibility that this could infect you and become a problem pretty quickly. One of the things our species has come up with to prevent infections like tetanus is by using vaccines. So when you're in your childhood, you can get immunized against a lot of different things. So basically what we get is kind of like a little bit of a dose of an inactive outer coating of a virus or a bacterium. And by getting this, what's going to happen is our body makes antibodies against it. So if we were to encounter it again over a span of months or years, your body will know exactly what to do in terms of its immune system response. So some of these are forever when we use vaccines, and some we need what are called boosters, because it's almost like your body has forgotten about them a little bit and the response won't be as good as if you've been within a couple years of that vaccine. So we might need booster shots for certain types of infections we're trying to prevent. Remember, metabolism is the sum total of all the chemical events inside of a living organism. And with bacteria, often we highlight oxygen use as another way of classifying different families of bacteria. And they have different requirements. For example, we may see a group of bacteria that are obligate aerobes. Now, the air is the part that kind of gives away what these bacteria need. So these need oxygen in order to reproduce and do what they do. Tuberculosis bacterium is one that cannot live without oxygen. This is also one of the diseases that can hit our species as well, and and take residence within the lung tissue, and we know there's a lot of oxygen availability there. We also have the family of obligate anaerobes. Now, when you see this AN in front, what that means is these are obligated to live without air, so they're unable to grow in the presence of free oxygen. Those Clostridium bacterial cells, they spore up when they lose that oxygen. It's only when they get inside of a body with that extra moisture and warmth where there's low levels of oxygen that they come and eventually emerge back as a living bacterial cell. Facultative anaerobes, the term facultative means that it can go either way. So these don't really require air, nor do they don't like air, they don't hate air. They can exist in both environments. E. coli are like that. So we have E. coli in our large intestine. It's a permanent residence there, low levels of oxygen in there. But we can also grow E. coli on a lab petri dish very easily, there's a lot of oxygen around. So facultative anaerobes are bacteria that can live with or without air and oxygen. We can also classify bacteria based on their nutritional modes. Let's look at a few. We have the autotrophic prokaryotes. Autotrophs can make their own food. So one of the types we have are called photoautotrophs. They're going to use solar energy to reduce carbon dioxide. So in other words, they're going to be basically adding electrons and hydrogen ions to carbon dioxide to make organic compounds like sugars within themselves. We also have photosynthetic, those are photosynthetic. We also have chemoautotrophs. Now, chemoautotrophs are going to oxidize inorganic compounds. So what they'll basically be doing is adding oxygen to compounds without carbon to obtain necessary energy. When they do this, they're going to basically reduce, by the use of extra electrons and hydrogen ions again, carbon dioxide to organic compounds, basically again build sugars within that bacterium itself. We call this being chemosynthetic. The first type we looked at was photosynthetic. We also have the classification of heterotrophic prokaryotes. So these are going to have to take in nutrients from the outside. First type we'll look at are what are called saprotrophs. So these are aerobic saprotrophs. So these are bacteria that use oxygen to decompose most large organic molecules into smaller molecules. In other words, they feed on dead things. So in combination with fungi, bacteria are one of the helping organisms that allows us to break down organic matter and recycle it back to the environment. They are always essential components of a healthy ecosystem. Prokaryotes can be free living or they can be symbiotic as well. So some are just kind of on their own or perhaps in colonies, and some live in combination with other organisms. For example, there's going to be bacteria in these root nodules here of this plant. And what that's going to do, these are called nitrogen fixing bacteria. Okay, so we'll call them N-fixing. And what they do is they take nitrogen out of the air that gets down into the dirt, and they provide it as a form of nitrogen for the plant. The plant itself can't do that without these root nodules. We also have some scenarios where we have bacteria, algae, and fungi living together in kind of a harmony when we look at some of the things that we normally talk about with the plant kingdom. So there's this very close association with plants and fungi that make the whole system work better in terms of what it does for each of the organisms. These interrelationships, there's three different types. One is called mutualism. And mutualism is when all parties that are interacting together all benefit from that interaction. So with the root nodules, the nitrogen fixing bacteria give the plants a nitrogen source, gives a bacterium a protective home, and it gives the plants a good way to get nitrogen. A commensalists live with other organisms, providing nutrients in exchange for protection. The most studied one is going to be from here in a lichen. So you have a fungus and an algae that work together in harmony, where one gets protection and the other one gets the nutrients from photosynthesizing. So it's a very nice relationship between the two that allows two different species of organisms to interact with each other. And there'll be bacteria intertwined throughout the two of these as well. So it's kind of like a harmonious grouping of organisms that makes everybody survive, and they work really well together. The parasites, this type of bacteria, they're going to derive nutrients from their hosts. So ultimately, some of our bacteria will be classified as this. They will damage or kill the host. It's just what they do. It's a working strategy for that particular species. These are ultimately the ones that we worry about the most when we talk about bacteria. So why do bacteria get such a bad rap? What makes them so dangerous? So if you look at these bacterial diseases here, syphilis, gonorrhea, chlamydia, strep throat, tuberculosis, whooping cough, anthrax, and on and on and on. These particular conditions and diseases are very scary to people. But why bacteria get such a bad rap is that 99% of the bacteria don't do anything bad to you. Okay, they're completely harmless to our species. It's just that these particular diseases are so significant that we worry about them quite a bit. And we go to extremes and we get antibacterial gels that kill 99.9% of anything on our hands. But what we're doing is we're also killing some of the good bacteria that normally goes to battle for us. So ultimately, bacteria do get a bad rap. Especially because there's so many of them, so many that live on you and in you, that unfortunately we go to extremes to be bacteria free when we don't really need to. So is there no harm in bacteria? Well, of course not. If we are hit with the wrong species, what's going to happen is ultimately it can cause very bad things to happen to us. But our species has worked hard over the past few hundred years to develop a lot of medications and a lot of defenses for us. So if we do get infected by bacteria, we tend to survive most of those infections. Now, one of the bad things that can happen when we are so aggressive with bacteria and we try to kill everything, for example, again, with our 99.9% bactericidal gels and cleansers we put on everything, is that we may be setting up for a problem later. Here's how that would happen. Let's imagine that we have a bacterial colony that normally is killed by an antibiotic. But we go ahead and we use some heavy-duty cleansers, and this particular bacteria randomly gets a mutation that allows it to withstand the onslaught that the antibiotic normally does to it. Instead, this is a survivor bacterium right here. Now, what's going to happen is all the rest of the bacteria that don't have the mutation, they're going to die. But this one bacterium here that knows essentially the secret to surviving against that antibiotic treatment is now going to pass on that resistance gene to all of its progeny at a later time. And it's going to form a resistant colony. And this colony is going to be completely unaffected by antibiotic use later. So a new antibiotic will have to be found in order to kill this. So this is what we call the beginning of a multi-drug resistant bacterium or bacterial colony. Now, we also have the idea of conjugation, one of the methods of genetic recombination, where we send out a conjugation pilus, which you can see here, to another bacterial cell who doesn't have that drug resistance gene to it. And if it sends it over here, not only is this particular microbe harmful, but now it's harmful and it's going to resist the antibiotic that normally kills it. So each one of its progeny now carries that antibiotic resistance gene, and it will just keep on doubling and doubling and doubling over time. And again, you have a new strain of bacteria that is also resistant to that original antibiotic, just due to some genetic recombination. Let's pit stop just for a second because we're going to switch over to studying about archaeans next. So archaeans, again, is the other domain in which we classify prokaryotes into. What we want to be able to do is describe the attributes of archaea. We want to explain how they're different from others in the domain bacteria, and we want to describe some of their unique habitats. So before the 1970s, we didn't really differentiate archaea from the bacterial grouping. But In the 1970s, a man named Carl Woese discovered that the base sequences of their rRNA differs from that of bacteria. So it doesn't seem like a big deal, a little bit of rRNA in the nucleolus, but we also found out that there were some other differences the more we took a look at them. For example, if we looked at their cell walls, we noticed that archaeans do not have peptidoglycan in those cell walls like we saw in most of the bacteria. The lipids of the Archaean group have glycerol attached to branch chain hydrocarbons. Now, normally in bacteria, glycerol will be linked to long hydrocarbon fatty acid chains, but suddenly these chains are looking very different with those within the Archaean family. We have a lot of branch chains and not a lot of elongated single chains. When we look at the archaeans biochemically, when we analyze what's inside of them, they're starting to look a lot more like eukaryotes than actual bacteria. Let's take a look at some of the characteristics of their metabolism. Most of them are chemoautotrophs. So again, what that means is they make their own food through inorganic compounds that they oxidize. Some are mutualistic, so they live in a close harmony or relationship with another species where they both benefit. Some are commensalistic. What that means is that ultimately one will benefit and the other one will just exist without having a benefit or harm. There's none to be known as parasitic, so none that we have found yet hurt our species or other species. None are photosynthetic, so that must mean that we're going to find them in places where light is really not to their advantage at all. And the really cool thing about them is they're found in some of the harshest conditions on Earth. And you can see from the picture down below, this kind of looks like a pretty inhospitable place. And we'll get more into that in just a minute. The types of archaeans that are out there, we said live in harsh conditions. Some of those conditions are very anaerobic, such as marshes or even intestinal tracts of living things. And we call this grouping of archaeans the methanogens. One of the things that they're going to produce is methane gas as a byproduct of their metabolism, and ultimately might lead to the smell or the flammability of gas pockets in a swamp environment, or even that environment of a large intestine of an organism. They'll produce methane from hydrogen gas and carbon dioxide. The second group is going to be called the halophiles. And the halophiles are going to live in very salty conditions. So these conditions are so salty, typically nothing else lives there. But they're going to require those salt concentrations for their growth. They have evolved special chloride pumps that bring chloride ions into the interior of the cell to prevent water loss. So they have found a way. Nature always finds a way. Hot sulfur springs, you'll find archaeans called thermoacidophiles. And these guys here are going to use sulfur as one of their nutrients and survive best at temperatures above 80 degrees Celsius. To put that into perspective, your body temp is about 37 degrees Celsius. So more than double your body temp is a great day, a great weather day if you're a thermoacidophilic archaean bacteria. They, from their name, acidophiles, means that they love acidic solutions to be surrounded by in their own personal environments. And the plasma membranes that they have contain unusual lipids that tolerate high acid levels. Here you can see a picture of some of those extreme habitats. We have halophiles, so you're going to find them living in this water that is so salty. You can see this kind of salty crust here where the water has evaporated on the shoreline. We're also going to take a look at where the thermoacidophiles live. So imagine areas that have a lot of hot springs and geysers, bubbly sulfuric water solutions. You'll find thermoacidophilic archaean bacteria in those places. And then lastly, we have those methanogens, and we said, where they produce the methane. In pockets, they'll be living in these anaerobic conditions down in the muck, and they'll bubble up those gases that they're famously known for. So we've reached the end of our study of bacteria. What we want to be able to do, again, be able to explain how we classify bacteria. We went through a number of ways today with our talk. Number two, what are those structural characteristics that are common to bacteria, and what do those particular structures do? How do they function? How do bacteria reproduce? Describe mechanisms of genetic change in bacteria. We listed and explained 3 different ways that you can genetically recombine and form essentially a new bacteria with new traits. Number 6, explain what spores are. 7, describe why bacteria get a bad rap. And number 8, explain the sneaky mechanisms that allow bacteria to survive. So again, reflect back when you're done with the lecture. Make sure that you have a good grasp of those things. And if you don't, read back over your notes, perhaps watch a segment or 2 again so you'll know that information a little bit better. I hope you learned a lot from this lecture, and I'll see you again next time. Bye-bye.